Hey Team Bio, welcome to your screencast, the introduction to the cell membrane. So we're going to move from talking about proteins and enzymes to talking about cells, what they're made of, and how they create a constant internal environment, how they maintain homeostasis. And a big way that they do this is by keeping the insides of them separated from the outside of them, and that is accomplished by the cell membrane. So the cell membrane is actually made up of all the big four organic molecules. So remember, our big four are fats and lipids, proteins, carbs, and nucleic acids. But the organic compound that plays the biggest role in the structure, the overall structure of the cell membrane, is actually a fat. So all of our cells are made up of just tiny little balls of fat. Okay, so I think that if you were in my class last semester, we talked very briefly in a screencast about phospholipids, and we're going to spend a lot more time talking about their structure and function in this screencast, but maybe you've heard this word before or seen this image. Okay, so this is a phospholipid. It is composed of two fatty acid tails here, which you should hopefully remember. A fatty acid tail is very nonpolar. Therefore, it's very hydrophobic. There's one that is a long straight chain. So remember, this is a saturated fat. There are no double bonds between the carbon backbone of the fat, so it's a long straight chain. And then this is an unsaturated fat. And therefore there's a double bond that creates a little bend in the molecule. Okay, this mixture of saturated and unsaturated fats within a phospholipid helps control the fluidity of the cell membrane. Remember, saturated fats are solid at room temperature and unsaturated fats are liquid at room temperature. So the combination of saturated and unsaturated helps determine the kind of melting point, if you will, of our cells. Okay, so this is one side of the molecule, the fatty side, and this side of the molecule is called a phosphate head. It's composed of a phosphate group and the glycerol, and it's very polar, which means it is strongly hydrophilic. Okay, so when we have one single phospholipid, it doesn't behave in a very interesting way. However, if we take a group of phospholipids and we throw them into a watery solution, they spontaneously form a structure called a bilayer. So if you take water, I'm going to make this a little bit bigger. So you take a bunch of phospholipids and you put them in water, what's going to happen is they're going to all align themselves such that the nonpolar tails face each other and the polar heads face the water. The nonpolar tails, as you remember, they're hydrophobic. So they do not want to be interacting with water. And the phosphate heads are hydrophilic, so they want to be touching the water. So this just happens spontaneously, the spontaneous formation of a structure called a bilayer, because it is the lowest energy state possible, where the hydrophilic things are touching water and the hydrophobic things are touching each other. Okay, but I'm sure you're asking yourself, well, what about this region right here? Won't the water flow here and then interact with the hydrophobic tails, which would be like a not great thing for them? They don't want that. And the answer is, you're right. This layer, this bilayer, is actually going to extend into a giant circle, which we call a cell, where the interior phospholipids on this side are interacting with a watery solution on the inside of the cell. And so this is water. And the exterior phospholipids in the bilayer are interacting with a watery solution outside of the cell. So we have water inside and outside, and then we have our lipid, our phospholipid bilayer, creating our cell. And like I said before, this just happens spontaneously. If you just throw a bunch of phospholipids into water, they will spontaneously form a bilayer that it kind of looks like a little bubble in the water. This image kind of gives you the impression that this is a two-dimensional structure, but actually this is very much a three-dimensional structure, which I think this image does a better job of showing you. We've taken a little slice out of it, but you can see we've got water on the interior, water on the exterior, and then here's our lipid bilayer. In this image, the phospholipid phosphate heads are red. They're right here, and they're all attached to fatty acid tails here. So we got one side, and then we have the other side of the bilayer here. Okay. So cell membranes are what we call semi-permeable. I guess I should say cell membranes. BR. They are semi-permeable, meaning they let some things pass through them, and they exclude other things from moving through them. And we're going to talk a lot more about what can pass through easily and what needs help getting into and out of the cell. So that's more on that to come. This idea that a cell membrane is semi-permeable, it's not going to allow certain things to pass through, but it will allow other things to pass through. Okay, and the last thing I want to talk to you about in this screencast is this idea of the cell membrane as a fluid mosaic model. So this image of the cell and this image of a cell give you the impression that the cell membrane is only composed of lipids, and that is not true. The cell membrane is actually studded with many different proteins and carbohydrate groups. So certain proteins form channels which allow small particles that couldn't pass through the cell membrane without some assistance. These protein channels allow things to move into and out of the cell when they are open. So this would be an example of a protein channel. Here we have another protein channel that's studded with a carbohydrate. Usually the function of these carbohydrate chains is to serve as like little molecular flags telling other cells what kind of cell this cell is. The other thing that might be present in a cell membrane is a signaling molecule. It's going to bind a substrate on this side of the cell, and then a piece of another enzyme inside of the cell is going to break off and go do something inside of the cell. So for example, if this is a hormone, we might be getting the signal to start making a new protein when this molecule is signaled to go do something inside of the cell. Okay, so that kind of gives you the impression of why we call it a mosaic. A mosaic is a group of tiles of many different patterns and colors, and our cell membrane is studded with a lot of different components, not just phospholipids. So that's the mosaic part. The fluid part of this model is this idea that nothing in the cell is fixed in one place. This phospholipid is actually beep-bopping around in the cell. And if we labeled it with some sort of, like, say, a radioactive tag, we turned this guy radioactive, we might notice that an hour later, this phospholipid has actually moved in the cell membrane, and now it is over here. So the cell membrane, things are not fixed. They are free to move around within the cell membrane, and they are moving. So that's the fluid part. Things are moving, and it's not just phospholipids, it's a whole mosaic of different components in the cell. Okay, that's it for this screencast. Big takeaways: that a bilayer spontaneously forms because of the hydrophilic and hydrophobic nature of a phospholipid. It will form a little cell structure, and it's semi-permeable. Certain things can pass through and certain things are excluded. We'll talk more about what can come in and what can't, or I should say go in or out, in our next screencast. And this fluid mosaic model, this idea that things are moving around in the cell membrane, they're not fixed in place, and there are lots of different components within the cell membrane, not just phospholipids. Okay, that's all.